Now that we've covered all the background material, we're going to start talking about specific techniques for plant propagation, starting with stem cuttings. Now you can see my silly little cartoon here. It's the free and fun way to fill your garden, and that's pretty much how I viewed stem cuttings when I first learned about it. Magical. I can just take stem cuttings of everything and get free plants. Well, it's not totally free. You do have to have media and containers and that sort of thing, but it's still probably cheaper than buying a full-size plant. So before we talk about how to take a stem cutting, we're going to spend a good amount of time on the science behind stem cuttings and why they work. So remember just a few things from our previous lectures. First, that cells make up tissues, which make up organs, such as roots, which is what we're trying to get to grow on our stem cuttings. And then remember these terms, totipotency, competence, and determinism. Do you remember what all those words mean? Well, if you don't, you should go back and review them. Especially totipotency, which that's why I have this picture here, just as a reminder for that one. Totipotency is the ability of every cell in the plant, or the fact that every cell in the plant contains all the genetic material it needs to form a completely new plant. That's why when we have a small piece of leaf in tissue culture, like in this picture, we can get whole new plants just from that one tiny piece. This is also why we can get roots to form on stem cuttings. They're all stem cells, but they still have the genetic information in them to become root cells. So stem cuttings, like any other type of asexual propagation, are possible because of vegetative growth. This makes sense because stems are a vegetative tissue. Now, vegetative growth is going to occur in what we call meristems. Now, meristems, you may remember from our discussion of plant anatomy and physiology when we talked about meristematic tissue. Meristems and meristematic tissue are the same thing, but these are areas of rapid cell division and they're growing points on the plant. Right? And that cell division that's happening is mitosis, which you'll remember is the type of cell division that gives us genetically identical cells. If you think about that, if you have a lot of genetically identical cells being produced, it would make sense that the plant that you're getting from that type of propagation in which mitosis is involved will be genetically identical to the parent plant. Right? It all fits together. So there are a lot of different places in the plants where we can find these growing points. One is at the tip of the stem, and there's sort of a blown up picture of that. One is at the tips of roots, in between the xylem and phloem, and in buds on the stem. All of those places are places where we can find meristems, right? And when we have these meristems that are actively growing and dividing, these can be places where we get the formation of new plant organs, such as roots on our stem cutting. Vegetative growth will occur from the meristems, and it can also occur in response to wounding the plant. So when you take a stem cutting, you're cutting it off the parent plant and you are creating a wound by cutting it there. And having that wound also stimulates vegetative growth in the plant. So we have vegetative growth going on in our stem cuttings, and within that category of vegetative growth, we have something very specific going on called adventitious growth. You'll often hear the roots on a stem cutting referred to as adventitious roots. So adventitious growth is defined for you here on the slide. It's when we have new tissue arising spontaneously in areas where it normally wouldn't. Right? So roots, for example, forming on a stem. You don't expect roots to form off of a stem, especially a piece of the stem you cut off near the tip of the branch. You expect them to form on other roots or at the base of the plant. You can also have adventitious shoots, and if you look at the picture here, the picture on the far left, that is a root cutting. So we had a piece of root tissue that produced both adventitious roots and adventitious shoots. Again, you don't expect for a shoot to grow straight off of a root. You expect it to grow off of a stem. So that's the picture on the far left. The second picture from the left is a stem cutting with adventitious roots. And the two stems that you see in the picture on the right, these are plants that have been wounded and they're getting ready to put on adventitious growth. A lot of times adventitious growth occurs as a result of a wound on the plant, and again, when you take a stem cutting, you've created a wound by cutting it off. Adventitious growth, because it is a type of vegetative growth, does develop via mitosis. Okay, so again, all the cells being formed, the new cells are genetically identical, and it's going to happen, this adventitious growth, on an already established vegetative structure such as a root, stem, or leaf. So let's dig a little bit more into the whole process of the formation of these adventitious structures. Again, they could be adventitious roots or they could be adventitious shoots. In the picture here, this is another picture of tissue culture, and those are actually adventitious shoots that have formed. Well, the whole reason why adventitious growth can happen is because of a process called dedifferentiation. Now, we've talked a little bit about differentiation before. Differentiation is what allows a cell to become a specific type of cell. So remember, all the plant cells have the same genetic information, but they will specialize into particular types of cells: root cells, stem cells, leaf cells, guard cells in the leaf, flower cells, all of that. Well, just as cells can become more specialized, they can also go back. They can become unspecialized. They can go from being a stem cell or a leaf cell back to just being an undifferentiated cell. So we call that dedifferentiation. To go back to our analogy of college students, this would be like if during your junior year you suddenly realize you don't want to be a political science major anymore, you want to be a horticulture major. So you dedifferentiate back to basically square one. If you haven't taken any horticulture classes, you're pretty undifferentiated at that point until you start taking your horticulture classes. I'm not trying to send you a subliminal message here at all. Okay, so dedifferentiation. It's the ability of cells, even in mature tissues, to return to the meristematic condition and produce a new root, shoot, or both. Right? The meristematic condition is that actively growing condition. When a cell has become very specialized, it's no longer meristematic. It stays what it is, and it doesn't actively divide that much. Now, the extent to which a cell can dedifferentiate varies. So this, again, it's much easier to dedifferentiate your sophomore year than it is your senior year, right? Same thing with plant cells. You remember we talked about determinism and how reproductive structures have a very high level of determinism in their cells. So a flower cell or a fruit cell, it can't really dedifferentiate enough to produce a new root or shoot, typically. What can dedifferentiate very easily are our parenchyma cells, which you'll remember are large, puffy cells with thin cell walls. Those parenchyma cells, just in general, are never very specialized, so it's very easy for them to dedifferentiate. So most of the time, our adventitious structures like roots and shoots are going to come from these parenchyma cells. And so the formation of adventitious roots happens in 3 steps. First, we have dedifferentiation, which I just told you about. Parenchyma cells become meristematic, they dedifferentiate and become unspecialized cells that are actively growing and dividing. Then, once they've dedifferentiated away from being parenchyma cells, they then differentiate into a new, different specialization. Right? They switch majors. And so in the case of stem cuttings, that differentiation is basically you have parenchyma cells in the stems that dedifferentiate and then differentiate into root cells. And that's step 2. So there's the initiation and organization of root initials, which are just sort of precursors of actual roots. And then the 3rd step is the development and emergence of the new roots. So in the picture here, these are some stem cuttings that I rooted. They had different treatments because they were part of an experiment, so they have different amounts and sizes of roots, but those are adventitious roots that have formed on stem cuttings. Funny thing about this picture, you can see that's not the best background for showing the roots. There was a old, almost retired professor around. He was not my advisor, he was just another one at the research station when I was in grad school. And he saw me trying to take pictures of these rooted stem cuttings for my research, and he said, oh, you should put them on the concrete and take a picture of them. It forms a really good background. The roots will show up really well. And I listened to him because he was really old, and I figured he knew what he was talking about. But I think, because he was kind of a mischievous guy, he was just messing with me to see if I'd just do it because he told me to. Because actually that's a horrible background. You can't see the roots very clearly. I should have used just a plain black background. So, I don't know what the point of that story is, but now you know why that picture looks like it does. And with that, you now also know how adventitious roots form on stem cuttings. Now, there are two types of adventitious roots that you'll find on plants: preformed and wound-induced. Some plants actually already have adventitious roots growing on them even before you take a cutting. They just naturally have these preformed adventitious roots growing on their stems. And a good example of this is our English ivy, pictured here. You can see the adventitious roots on the stem. It uses them to climb, but it also makes this plant really easy to propagate by stem cuttings because it already has roots on it. You don't even have to wait for them to form. You just cut off a piece, plant it, and you have a new English ivy plant. This is also one of the reasons why English ivy spreads like the dickens. Some plants have preformed adventitious roots and they're not as showy. They may not even be that visible. They may just be those root initials, the root precursors below the bark. But plants that have them, again, are very easy to propagate by stem cuttings. So hydrangeas, for example, have preformed root initials. Willows have preformed adventitious roots. So that's one type of adventitious root. The other type is wound-induced. These are plants that don't already have that, but when you create a wound by taking a cutting, by cutting it off the parent plant, it will then produce adventitious roots. Now, the way this generally happens, sometimes they'll just form the roots directly, but a lot of times when you wound the plant, it will first form what's called callus. And callus is just an irregular mass of parenchyma cells. So it produces a whole bunch of parenchyma cells because those are the ones that are going to de-differentiate easily and become our new roots. The callus, if you look at it, I don't have a picture of it here, but it usually has a whitish beige coloration and it's somewhat hardened, but the cells are pretty loose, and again, they de-differentiate very easily. And then once they do, the new roots form. So for wound-induced adventitious roots, again, typically you have callus formation first and then new root initiation. And this may be something that you observe on your own cuttings. You might see a whitish mass forming at the base of the cutting. That's okay, that's just callus. In the next few mini lectures, we'll talk about the environment you need to keep your cuttings in, the different types of stem cuttings, and then how to actually take one.